All right, welcome back, everybody. We are going to be starting tissues today. So we're going to learn about different types of tissues. We'll talk about some of their origins, and really the whole idea is the structure and function of different types of tissues. There's going to be 4 different types of tissues. Their structure's all different, which means they're going to function all differently. We'll discuss how cells of tissues are held together, because remember, cells come together and make up tissue, so they have to be these different types of junctions that hold them together. And then we'll compare these 4 different types of tissues: epithelium, connective, muscle, and neural tissue. Of these 4, epithelium and connective is a little bit lengthy. Muscle and neural tissue is always a lot easier and more fun to learn. But epithelium and connective is extremely, extremely important, and you'll understand why from a nutritional perspective soon enough. We'll talk about different types of membranes, and then we'll understand a little bit about tissue repair. So let's talk about different types of tissue. So remember, a tissue is a group of cells that usually have a common embryonic origin in an embryo and functions together to carry out specialized activities. And there are 4 basic types of tissues in the human body, and they're categorized like everything else, right, based on their structure and function. So the first type of tissue is going to be epithelium. Now, what's unique about epithelium, when we look at this right here, you'll see that under a microscope, there are some unique characteristics that differentiate this structure to connective tissue, muscle, and neurological tissue. Under a microscope, the cells of epithelial tissue are held very, very closely together with very little extracellular substance. Whereas if we looked here at connective tissue, you have cells scattered around, but lots of extracellular substance. So each of these, if you go to epithelium, this is a cell, right? And that's the nucleus. And then right next to it, very tightly bound, is another cell, and then another cell, and another cell. So all these cells come together to make this type of tissue. Whereas connective tissue, well, here's a cell, and then over here is another cell, and over here is another cell. They're not so held closely together, and then there's lots of extracellular substances around. So that's, under a microscope, a very, very clear delineation between these two. Okay, so I want you to understand that. And then there's many subclasses of epithelial that we will talk about, and we name epithelium based on the number of layers. So if we see that it's only 1 cell layer thick, which this is, we would call it simple. Okay, and if we had many layers, if there was another layer above that, like this, and it had many, and then there was another layer above that, like this, then we would say that this is stratified, okay? And because these cells have this tall column type of shape where they look like buildings, then we would call them, if it's 1 cell layer, we would say it's simple columnar epithelium. And then if the shapes were more like cubes, like this, and they were closely packed together, then I would call it simple cuboidal epithelium. And if they had different shape, and if they were flat and kind of irregular shape like this, and again it was only 1 cell layer thick, then we would call it simple squamous epithelium. And then if it had multiple layers, then we would call it stratified squamous epithelium. If it had multiple layers of these cubes, then we would call it stratified cuboidal, okay, epithelium. Connective tissue has a whole different category system. We'll go through that shortly. When we talk about muscles, 3 basic types of muscles. There is skeletal muscle that attaches to skeleton, and it is voluntarily moved. And then we have smooth muscle, which is located around your organs and your glands and your blood vessels. And then we have neurological tissue, which really is the neuron, and it's electrically excitable. The other type of cell that we have in the neural system are called neuroglial cells. But the neuron, which is what we're seeing in this picture, is the main type, and it has a few key parts that we'll talk about. These branches that we see over here, and these branches, these are called dendrites. In Greek, dendra means tree, and this cylinder right here, that's called the axon, and then this structure is called the cell body, and the cell has many of the organelles that we've already learned, right? You see that there's a nucleus there, we've got some mitochondria in there, we have endoplasmic reticulum and ribosomes to help make proteins. So 4 basic types of tissues, you should know them: epithelium, connective muscle, and nerve tissue. And the next hour and a half to 2 hours, this is going to probably be a long lecture, we'll be talking about the different characteristics and where they're located, okay, and what their functions are. Now, in order to make tissues, what do we need first? You got to go back to the organization of living. We would have to say chemistry makes up cells, and then what? Cells make up tissues. Exactly. So let's look at how some of these cells can come together. When they do come together, they come together by these way of cell junctions. Let's take a look at a short little video on this.
The cells of many tissues are joined together by intercellular junctions that are located between the adjacent cells. Most types of epithelium and some types of muscular and nervous tissue contain intercellular junctions. There are 5 types of intercellular junctions: tight junctions,
very important,
adherence junctions,
tight junctions are important,
desmosomes,
so with desmosomes,
hemidesmosomes, gap junctions.
Gap junctions, very important.
The tight junction is an area where the plasma membranes of adjacent cells are fused together. These junctions protect underlying tissues from harmful substances by restricting the movement of substances, like hydrochloric acid, in the spaces between cells. Tight junctions are found in tissues lining the surface of organs and body cavities, such as the stomach, intestines, and urinary bladder.
Intestines, keep that in mind, tight junctions.
Adherence junctions are created when transmembrane proteins, anchored to a dense plaque of proteins inside the cell, connect the adjacent cell membranes together. Additionally, microfilaments extend from the plaque into the cytosol of each cell. The dense adhesion belts help resist separation of epithelial tissues. The desmosome is similar in structure to the adherence junction, except that intermediate filaments extend into the cytosol of each cell. These junctions help provide stability to tissues and are abundantly found in the epidermis of the skin and between the muscle cells of the heart. The hemidesmosome, or half-desmosome, provides strong attachments between cells and other extracellular materials, such as the basement membrane of epithelial tissues. Hemidesmosomes are most abundant in tissues that undergo constant frictional or abrasive forces. The gap junction is an open communication channel. Transmembrane proteins, called connexons, join together to create tiny tunnels that form gaps between the adjacent membranes. Gap junctions allow ions and other small molecules to pass freely from one cell to another.
And quickly.
Gap junctions also allow electrical impulses to pass rapidly from one cell to another.
Rapidly. Muscle
Gap
and
junctions
nerve.
are particularly abundant in some types of muscle tissue.
Muscle and nerve, very important. Okay, let's get back here. So the tight junctions, I think, are extremely important. They all are, but from a nutritional perspective, the tight junctions you're going to see frequently, and it's something that we work with pretty much all the time. It does create a watertight seal. It prevents the passage of water and solutes into parts of the body where we don't want them. Common in the GI tract. Okay, so in the gastrointestinal tract, the bladder, very important. We don't want certain acids and enzymes or waste products leaking. So if, just for simplicity, let's say here's a healthy part of the intestine, the tube, and then let's say here's another part. What I'm doing intentionally is I'm making it permeable, right? I'm making these gaps so that here in this one, the top one, we want food and enzymes staying in here. But there are things that can actually damage the epithelium so that these food particles and these small proteins can actually leak and make it through and get into systemic circulation. These, let's say, are blood vessels. They can enter circulation or enter the blood vessels, and the body doesn't like that because now we have this protein that's larger than what it should be escaping, entering the blood vessels, and it could trigger an immune response that is typically referred to as intestinal permeability or leaky gut syndrome. Just some general information on that because I believe on the discussion board this concept may be coming up. Okay, but when these proteins escape and they get into circulation, the vibration of these proteins can mimic other proteins in the body, like the protein in the thyroid or protein in the joints. And the vibration of them, we call it molecular mimicry. Think of it as like a drive-by shooting, where there's a lot of innocent bystanders that get hurt. Or you think of, you're walking at the beach or you're walking at the mall and you think you know someone in front of you, you tap them on the shoulder and go, "Hey, Mike." And they turn around and it's not Mike. "Oh, I'm sorry, I thought you were someone else." The immune system can work the same, where it's trying to target a specific protein in that food, and it mimics the same protein in the cartilage in the joints. And when it attacks that hyaline cartilage, maybe that's referred to as rheumatoid arthritis. Or maybe it attacks the thyroid and you get a shutdown of that thyroid. Okay? Adherence junctions, these hold epithelium together. They're important, but we're not going to talk about it in too much detail. I think that we can focus on some of these other junctions, like desmosomes, I think are extremely important. These are found in the superficial layer of the skin in the epidermis. It's very, very strong. They're held tightly, and anyone that's ever had a sunburn that, can see that sometimes that top layer can peel off as a whole sheath. Okay, so that is desmosomes. They resist cellular separation or cellular disruption and twisting and compression. The heart has some desmosomes as well because that heart is beating pretty aggressively. We don't want heart cells tearing apart. And heart cells also have gap junctions as well, which is extremely important in rapid communication from one cardiomyocyte, right? Myo is muscle. Whenever you hear cyte, C-Y-T-E, you're thinking cell. So a cardio, heart, myo, muscle, cyte, cell, one to the other, they have to communicate rapidly so that all the cells in the right atrium and left atrium contract at the exact same time. And then all the cells of the right and left ventricle contract in sync as well. It's only because of those gap junctions that allow for that. Let's see here. Gap junctions, these are the tiny spaces between the two cells. They're crossed by these protein channels that are called connexons. It sounds like connection. We find them in muscle cells and nerve cells that allow for very fast, rapid communication from one to the other. They're found in the heart, they're found in smooth muscles, and they're also found in nerves. Okay, let's compare epithelium and connective tissue. So if you were to cover this up and you weren't looking at what it says on the bottom between A and B. Remember I said earlier, one of the biggest differences between epithelium and connective tissue is that in epithelium right here under a microscope, look at how tightly bound those cells are, right? Here's one, here's one, here's one, here's one, here, right? They're all like, it looks like someone's building a wall, right? Down here, these look a little bit flat and irregular, so I would probably say these are squamous cells, and because there's multiple layers of them, right? There's a lot of them here. I'd say there's stratified squamous epithelium, okay? And then off here on this side, we have some cells. Here's one, and here's one, and here's one. But they're certainly not tightly bound together like they are in epithelium, so that would make the one on the right connective tissue. But there's lots of ECM. I want you to keep that in check. ECM, extracellular matrix. Really important. When that extracellular matrix is healthy, the individual is healthy. When the ECM starts to degrade and break down, we get inflammation and chronic inflammation, and ECM integrity is very, very important. There's a lot nutritionally that can be done to maintain connective tissue as well as epithelial tissue. So let's look at epithelium tissue first. These are cells that are arranged in sheets, that they're densely packed. Many cell junctions are present. The epithelium is attached to a basement membrane. And, think of it as just like attaching cells to cement, right, to some sort of glue. Epithelium is avascular. When you see the word avascular, we can see that here's vascular. That means blood vessel. And whenever you put A in front of it, it means without. Avascular. Epithelial tissue doesn't have blood supply, but it does have nerve supply. And mitosis or cellular division occurs frequently, and that would be important, right? Because these epithelium are exposed to the outside world, right? Like your skin on the outside is epithelium. It's attached to the outside world. But interestingly enough, so are most of the tubular systems and tracts that are found inside the body because it's exposed to things from the outside world, right? So the mouth, if my lips are closed, right, you think it's inside the body, the esophagus inside the body, stomach, intestines inside the body, but yet it's exposed to things that I'm consuming from the outside world and inviting it into the body, right? A lot of foreign proteins going in there. If I swallow a cracker, a square cracker with a sharp edge to it, and I don't chew it and I inhale it, I can scratch the esophagus. Right, so that needs to, if it gets scratched, just like a scratch on the skin, those cells have to divide, multiply, replicate, repair, and heal. All right, so the intestines, although it's within the body, it's considered like a tube within a tube. And because it's exposed to things outside of the body, we would consider it to be epithelial tissue. Some of the functions. Physical protection. It protects exposed and internal surfaces, as we just mentioned, from abrasions, dehydration, and destruction from chemical agents, just like the stomach having lots of hydrochloric acid. That could burn, can it? And we know that many people it does that experience heartburn because that hydrochloric acid is very low on the pH scale. We think it's about 2 to 2.3, very acidic. Absorption, the lining of the gut and the respiratory tract allow for nutrients to be absorbed from the gut, and the exchange of gases between the air in the lungs and the blood. So when we talk about the lungs and we look at oxygen exchange and O2 and CO2 exchange, we're going to see that, well, maybe the type of epithelium that we find in the lungs may be a little bit different than the type of epithelium that you find in certain glands or sweat glands or mammary glands, or even in the stomach, okay, or in the kidneys. Excretion, the unique lining of the kidney tubules make the excretion and concentration of the excretory products in the urine even possible. It's involved in producing secretion. So glandular epithelium is adapted for secretory activities such as pushing out or secreting hormones, sweat, mucus, digestive enzymes. Epithelium is involved because it has a good nerve supply for providing sensation. The sensory nerves of the eyes. We know that if something hits the eye, bam, right away it closes, right? If you're outside biking and a little fly hits your eye, it shuts quickly. There are rods and cones in the eye as well for vision. There are receptors in there for color or for black and white. The ears are great for sensation. The skin, thank goodness, right? If you have something crawling on you, you could feel it. You slap it to try and make sure it doesn't sting you. And then there's some specialized epithelium, microvilli and cilia. When we look at different types of epithelium and you look at the name, it may not say just simple columnar epithelium. It may say simple ciliated columnar epithelium or pseudo-stratified ciliated columnar epithelium. So when you go to the apical surface, which means you look at the top surface and you see little hairs and little villi over there, then we can say that it's ciliated. Or sometimes you can actually see much, much smaller than cilia and very fine. It looks like a brush border, like a broomstick, the end of a broomstick, those bristles, microvilli that are used for absorption and secretion. Microvilli are designed to increase surface area, so we'll see lots of microvilli within the intestines. Cilia, on the other hand, like in the trachea, those are beating rhythmically upward to try and push debris up and out of the trachea, the windpipe, and out of the body. Otherwise, it gets deeper into the lungs. And we know that smoking can paralyze those cilia. So here is the apical surface, right? I said go to the top, and sometimes you'll see these small little cilia up at top here. So they'd be ciliated. This right here is epithelium, right? The cells are packed closely together, and you can see the nuclei there. And then here's the basement membrane. That's the cement. And you can see that epithelium and connective tissue are connected by this cement, by this basement membrane. So these cells are held closely together. And down in here, in connective tissue, well, here's a cell, but then, maybe a while till you see another cell. They're not held closely packed together. Here's the classification system that I mentioned earlier about epithelium. We said that they can either be simple, which is what we see here, one cell layer thick, or it could be stratified, where we have many layers. Okay. Now, this, you see the word stratified, but here it says pseudo, meaning it looks stratified, but it really isn't. And what gives it the appearance of being stratified is that here's a nucleus at this level, and these nuclei are at this level. But then these nuclei here are at this level, and then these nuclei are at this level, right? So, and you can see it here too. So it looks like it's multiple layers, but in reality, it's still just one cell layer thick. So it's called pseudo-stratified. Okay, this is real stratified. This one is pseudo-stratified. Okay, so one of the ways that we can name epithelium is by, let's look at the cell layers. That's one of the qualities or characteristics. And the second is, what does the shape look like? Is it cube-shaped? We'll call it cuboidal cells. Are they tall and column-shaped? We'll call it columnar. Or are they flat and irregular shaped cells? That's squamous. So we can have simple squamous epithelium, you can have simple cuboidal epithelium, and you can have simple columnar epithelium. You can also have stratified squamous epithelium, stratified cuboidal epithelium, and stratified columnar epithelium. The covering and lining epithelial tissue, the name of the specific type of stratified epithelium depends on the shape of the apical cells, right? You always got to go to that top layer to make that determination. Let's look at this brief. Oh, we can't. What's going on here? Okay, we'll come back to that later. All right, so let's go through some of the characteristics of simple squamous. So again, let's take a look and look at the word simple. So you got to ask yourself, well, let's go the other way. Let's look at this first and say, all right, how many cell layers are it? Well, this is only one cell layer thick. So because it's only one layer, it's simple. And then we go, all right, what's the shape of the cell? Well, this does not look like a cube on this microscope. It's very flat and irregular shape. So that's squamous. And its last name is going to be epithelium, simple squamous epithelium. So it's single layer of flat cells. We'll find these in blood vessels. We'll find these in the chambers of the heart. We'll find them in cavities, right? We remember cavities from the first module. And in the pleura, we're thinking lungs, pericardium around the heart, peritoneum around the digestive organs. Because it's very thin, it's great for diffusion, to allow certain ions to move back and forth, certain gases to move back and forth, water and liquids to move back and forth, and it's great for filtration. Okay, and another unique quality is that the nucleus, you can see that they are centrally located. And that's different. That's different when we look at different types of connective tissue as well. Okay, so it's great for gas exchange, so we'll see them in the alveoli of the lungs. It's great for absorption, and when you think of digestion, what are we doing? We're breaking down the foods. We want to absorb the nutrients and bring them into our tissues, and then we want to get rid of the waste product. And then in the kidneys, we have a structure called the loop of Henle in the kidneys. And in the kidneys, there's a bunch of tubes, and a certain component, a certain aspect of the nephron, which is the functional unit of the kidney, is an area called the loop of Henle. And let me draw this out for you up here. Let me just clear this out. Let me go back to it. So this is just a very general drawing, just to give you an idea, because when we say loop of Henle and tubules, you should Google that and look at it in your textbook as well. But let me see if I can simply and quickly draw something out. This is called a nephron, and a nephron has a few parts to it. So it's going to have a very wide blood vessel coming in here, and then a small one exiting out. And then let me change the pen color here. And then we have something that looks like this. And then we're going to have something that looks like this. And we'll come out this way again, and we'll make this convoluted like this. And then we'll do that. Okay, so blood comes in here through this afferent vessel. Afferent means bringing to, efferent means taken away. So blood's coming in through this afferent blood vessel, or afferent arteriole, and then it's exiting an efferent arteriole. And as the blood is coming in, that blood is going to be filtered the blood is going to be filtered in here, and this is called the glomerulus, and it pulls out filtrate. It pulls out waste products, right? Because blood comes in, it's getting filtered in the kidney. This nephron's in the kidney. It's the functional unit of the kidney. And a lot of the clean blood is going to be returned, but the filtrate is going to move in this direction. And this convoluted tubule right here is called the proximal convoluted tubule. And if there's a proximal convoluted tubule, there's going to be a distal convoluted tubule. And then between is going to be this loop. That's the loop of Henle. Okay, so what's going through the proximal convoluted tubule and distal convoluted tubule loop of Henle is filtrate, which eventually that's going to push out and produce urine. Okay, so that's just general information right here. When you see that loop of Henle, at least you have a general idea as to where it is. It's part of the nephron, and actually this loop, if you have any relatives or friends or family members that are on blood pressure medication, many of them are on diuretics, and I believe one of them is called Lasix, which is a loop diuretic. And where does that diuretic work? In terms of pulling out water at the loop of Henle. That's why it's called a loop diuretic. Stratified squamous epithelium. You can look at this picture right here, and you can see that based on this illustration, stratified squamous, you can see that it's multiple layers here. It's not just one layer. It's stratified squamous. So any area that's exposed to irritation or enzymes or that you could damage easily, well, you can walk and scuff your skin against a sharp edge of a table. So the surface of the skin. When you're consuming food, you could bite your tongue, bite your cheek. So the lining of the mouth, the throat, esophagus, the rectum, anus, and vagina. These are all stratified. You need multiple layers because if it was just one cell layer and you scuffed your arm, you'd bleed right away. It's got to go through all those layers of epidermis and then the 5 layers of epidermis and then the 2 thick layers of the dermis and then the hypodermis, right? So anyway, that's stratified squamous. Here's simple cuboidal. So when you look at these, again, it's 1 cell layer thick. They're cuboid-shaped cells. And I'm really glad I drew out that picture before of the kidney and the nephron because look where the simple cuboidal epithelium is found. At the proximal and distal tubules. So very important. It's going to be able to filter different types of substances. So if it was simple squamous, the filtration there is going to be different than what's being filtered here, simple cuboidal epithelium. So it's great for absorption and secretion. So you can see, oh, there's my picture. How nice. Okay, so here is the kidney, and here's like an afferent arteriole, there's the efferent arteriole, and this is the glomerulus, and then that right there is the Bowman's capsule, and then this right here is the proximal convoluted tubule, and then this is the loop of Henle, and then you have the distal convoluted tubule. And right here, these cells are 1 cell layer thick. They're cube-shaped, simple cuboidal epithelium. Under a microscope, you would see this. Okay, and it's important to be able to identify these because I know on an exam I'm probably going to have pictures and you're going to have to identify them. This is different. Look, it looks like it's cube-shaped here. This is a gland. Once I see these multiple layers of cuboidal, now it's stratified cuboidal epithelium. These are going to line some ducts. Okay, ducts are usually linked with certain glands. So in this case, it's a sweat gland. All right, so these are kind of rare, but they exist. Here is simple cuboidal once again. When you look at this slide, this space here is the lumen. This space is the lumen. So this is now apical surface, and this is the other apical surface. So when you look at the apical surface, whatever this is right here, this could be the kidney tubules. That means that right there, this is the apical surface, simple cuboidal epithelium. When we look here and we look at the top, well, there you go. Look right at the apical surface. Here's cilia. These are tall shaped cells, so these are columnar. They're one cell layer thick. So it's simple columnar epithelium, but it's ciliated. A very important structure is this called goblet cells. These goblet cells are going to release mucus to the apical surface. So it's very important to have mucus in the respiratory tract so that debris can get trapped in the cilia and then it pushes it up and out of the body. Also, these cilia are found within the uterine tube. So those cilia beating rhythmically helps to push that ovulated egg right through the fallopian tube or the uterine tube. Simple columnar epithelium. Now, you can tell that this layer here, since the majority of these cells are in one layer, it's not pseudo-stratified, with the exception of this little guy here. Let's take that one out. This is definitely simple columnar epithelium. And then these are the nuclei to the right. You can see them here. Pretty much in alignment, right? With the exception of that bad boy right there. So simple columnar, these are going to lie in the stomach, the intestines, the gallbladder, certain parts of the uterine tube, the collecting ducts of the kidney, and again, protection, secretion, and absorption are their functions. Under the microscope, again, you would say, well, here is the lumen of the uterine tube or the fallopian tube, and then that's the nucleus. These are cilia, and these cells are column-like. They're columnar, so we'd call it ciliated, simple, columnar. Stratified squamous epithelium, several layers thick, right? When you look here, flattened squamous surface cells at the apical surface, there's your basement membrane connecting it to the connective tissue. I'm thinking skin and epidermis, okay? It's tough and it's water resistant. The skin is not waterproof, the skin is water resistant. We know that it's not waterproof because when you go in the pool and you go in the ocean, you can see different things happening to your fingertips. They can swell, they can dehydrate, they can shrivel. So keratinized are the surface cells at the top that are dead and they're filled with this protein called keratin. Non-keratinized, these resist abrasions but will dry out if not kept moist. The non-keratin in most living cells are at the surface of the mouth and the vaginal region. Here's a classical illustration. This is a microscopic section of the vaginal region, and you could see many layers, let's say from here to here, here to here. Those are squamous, stratified squamous epithelium, many cell layers thick. All right, so now we're talking about the vaginal region, we're talking about the fallopian tubes, uterine tubes. When women go for a pap smear, and maybe this is something many of you didn't know, but pap comes from Papanicolaou smear. George Papanicolaou was the physician. And they're collecting sloughed off cells from the uterus and the vaginal walls. And it's designed to detect certain types of dysplasia or changes to the shape of the cell. When women go for a pap smear and they say there's cervical dysplasia, it just means that the shape of the cells, they're changing and they're not all symmetrical. It should be done annually for women over the age of 18 or once sexually active. This is some scary statistics. HPV, human papillomavirus, is seen in 80% of women over the age of 50 have it. And usually asymptomatic, which is how most diseases are spread, right? When someone doesn't know they have it, that's how it's spread. And HPV comes in different flavors. There's low risk and high risk. The low risk typically shows up as cervical dysplasias, irregular cells, and then high risk are the ones that lead to cervical cancer. Cervical cancer is an STD. It starts off as HPV and then it kind of migrates towards that. The reason why this is important, nutritionally speaking, is many young women who are on the pill or the oral contraceptive, there is a DIND associated with every drug. So what is a DIND? A D-I-N-D. DIND is a drug-induced nutrient depletion. So one of the first things I do in my office when I look at an intake form is I'm always looking at the medication list. And within the first, it's not the medication list, for in many instances, it helps to determine the safety of the care that I'm going to provide them, because there are certain patients that I may not be able to treat or take care of depending on the medications that they're on. But there's also drug-induced nutrient depletions. So that means that by taking the drug, it's pulling out and depleting certain nutrients. And if you deplete it of certain nutrients, a lot of biochemical reactions can take place and your physiology becomes impaired. And when your physiology is not functioning, you get pathophysiology or disease. So with the OC, or the oral contraceptive, it commonly depletes B6, 9, and 12, especially B9, which is what people refer to as folic acid. And when you're folic acid deficient, there's lots of research that shows that you can get cervical dysplasia. And simply by supplementing with the right form of B9, many people cannot take B9, but they need to take it in the form of 5-methyl tetrahydrofolate or 5-MTHF, then within just a few months of time, their folic acid levels come right up and the cervical dysplasia dissipates. Okay, but it's also why women, when they're on the oral contraceptive and they come off and they want to get pregnant, they shouldn't get pregnant right away because their folic acid levels, B6, B9, and B12 levels are kind of depleted. So you want to make sure that you just don't want to run a marathon. You want to train for it. So pregnancy should be the same. You want to train for that and practice for that and make sure that you are fully equipped to maintain a healthy pregnancy. Gardasil only protects against 4 out of the 10 strains, and it's very, very questionable. There's lots of drug-induced nutrient depletions and many people, unfortunately, who have been hurt from that type of vaccination. And I'm sure you've seen them all over, YouTube and all over Facebook. These videos are out there, and they've made their rounds. Transitional epithelium. Okay, when I see transitional and I think of transitional, I primarily think of one area, the urinary bladder. Transitional because the urinary bladder changes in shape depending on whether there's urine being stored or not. So think of a balloon. It's got one shape. When you blow it up, it stretches. So what's going to happen to the cells? They're going to change in shape as well without damaging. So when you see a variety of different cell shapes, I'm thinking transitional. Pseudo-stratified. Here's that classical representation of column cells. Let me change the color here so we can see it a little bit better. Let's make it blue. So here, here, here. These are still one cell layer thick, but they're pseudo-stratified. They look multiple because the nuclei are at so many different varying depths. Pseudo-stratified columnar epithelium, respiratory tract, male urethra, and the epididymis. The epididymis is part of the male urethra. Okay, and again, when you look down at the bottom, goblet cells are there. Goblet cells. These are cilia, goblet cells, pseudo-stratified because the nuclei are at different levels. Glandular epithelium. derived from epithelium cells that sank below the surface during development. When we think of different types of glands, I think of two primary classifications, either exocrine or endocrine. Exocrine is going to be a cell that secretes enzymes or a certain type of product to the epithelial layer, like outside to that free surface. Sweat, tears, milk, earwax, saliva, and even digestive enzymes. Remember I said, even though the stomach is in the body and the intestines are in the body, the digestive enzymes are being released in that lumen of the intestines, and that is exposed to substances that were at one point outside of the body. So exocrine glands. Endocrine glands, these are ductless glands, like the thyroid gland, the pituitary gland, the adrenal gland. These aren't releasing substances to the epithelium or outside of the body. It's secreting them into the bloodstream so they can travel around. These hormones are involved in maintaining homeostasis. If there's an issue, like Sjorgen's syndrome is an autoimmune condition of the exocrine glands, and these people have dry eyes, they have dry mouth. They'll typically say when they get up in the morning, difficulty in opening their eyes, and they have to like moisten their lips before they open up. So epithelial tissue, simple squamous, simple cuboidal, simple columnar. Stratified squamous, stratified cuboidal, stratified columnar. In terms of pseudo-stratified, this one only comes in one flavor, and it's the pseudo-stratified columnar or pseudo-stratified ciliated columnar epithelium. Okay, transitional can kind of go in all directions. Maybe at one layer it's starting off as columnar, and then it's starting to flatten out and it becomes cuboidal, and then it becomes very flat shaped, or you'll see a variety of all three of them. Glandular epithelium, the secretions of endocrine glands enter the interstitial fluid and diffuse into the bloodstream. The secretion of the exocrine glands enter ducts that empty onto the surface of the covering lining of epithelium, such as the skin or within the GI tract. Okay, so we did this already. These are just some other pictures, so you can certainly take the time to go through them, but this is repetitious, but I did want to put these illustrations in a variety of ways. Structural classification. They could be unicellular, like goblet cells that we saw, or they're multicellular. Sweat glands, oil glands, salivary glands, those are multicellular. Uni, the goblet cell. You can take a look here, all the way on the left, the simple tubular. Then we have acinar cells. If you look at D and E, simple acinar and simple branched acinar, we're going to see these acinar cells when we talk about the pancreas. And compound acinar, just different types of shapes, but all secreting a different type of secretion onto the epithelial surface. From a functional classification, we have mimicry, apocrine, and holocrine. So if we look at mimicry, apocrine, and holocrine, they're each slightly different. In the mimicry secretion, you could see the secretion is happening through what method? What is this called? Exocytosis. In mimicry, you see that part of the cell becomes pinched off, right? You can see here's the nucleus, you could see the endoplasmic reticulum, you could see a Golgi complex, and then part of that cell pinches off. So this would be like milk, like a mammary gland. The salivary gland, that would be your mimicry secretion. Whereas the holocrine secretion, this is when the whole cell actually dies and ruptures and becomes the secretion. That's found in the skin like the oil glands, and that's why when someone has oily skin or they don't wash their skin very well and the entire cell dies and ruptures and stays on the skin for long periods of time, it can lead to acne. Okay, I'm going to pause here, and then we're going to continue with connective tissue. I want to give you a little break.